Hello, and welcome to the Molecular Bio Chapter 15 review, where we will be going over capping and polyadenylation. Exciting! So it's not as cool as splicing. That was my favorite chapter, but capping and polyadenylation is cool too. So I'll have the Quizlet up here, and we'll go through the PowerPoint at the same time. So the important thing about polyadenylation, capping, and splicing is that they are co-transcriptional, which means as the mRNA is being made, splicing, capping, and polyadenylation are happening. Later on in the chapter, we learn about how the polymerase II C-terminal domain is going to aid in facilitating that through its different phosphorylation states, but I don't want to get ahead of myself. The main point just to remember is that the splicing is the capping, the splicing, and the polyadenylation are all happening, starting with your capping, and then you get your splicing, and then your polyadenylation at the end. All right, so what is capping, and when does it happen? So capping is this process by which we get this the M7G cap on the end of your mRNA. When it happens, it occurs early on in transcription when the 5' end of the RNA first emerges out of the back of the polymerase. So if I was to pull up a picture of transcription direction, there. So we have this 5' strand here, and this is what first starts sticking out. The RNA itself is being synthesized on in the 3' direction. So the very first thing that's kind of going to be like shot out the butt of the polymerase is the 5' of this RNA. So when it starts to stick out, like the first little bit of it, about 30 nucleotides, that's when you're going to have capping happen. So there is something that's notable about the cap is that there's a lot of methylation around the cap. And when we take a look at the structure, this is a messy picture that Dr. Doud drew on when I was taking her class, but I'll break this down. It actually makes sense. There's methods to the madness. So up here on the top is your cap. So we know that it's 7mg, and what that stands for, or M7G, what that stands for is 7-methyl-guanosine. If we know about the structure of a nucleic acid, right, you have your phosphate group, your sugar backbone, and then your nitrogenous base. Here is the nitrogenous base guanine. Right here is our ribose sugar with the R. And then these three things here, this is your phosphate groups. So if we look at this guanosine residue, if we look right here in the corner, it's very hard to see, but these are numbered. So each of the elements, I guess there's nitrogens and carbons in this ring, they're numbered, each position. So this little N in the corner here, this is the 7th position on this guanosine. So on the 7th position, there's a methyl group. So that's why it's called 7-methyl-guanosine, because there's a methyl group on the 7th position in this double ring structure. So then it's just hooked up normally to your ribose sugar. What's also notable is that there's a triphosphate linkage between the cap and the first nucleotide. Normally, we know that there's phosphodiester bonds between nucleotides. That's common sense at this point, or common knowledge, sorry. And in those phosphodiester bonds are a single phosphate group linking them, but we can see 3 here, so that's a bit unique about the cap. So we can count them, 1, 2, and 3. You can see these 3 phosphoruses here. So this then is nucleotide 1, which just shows the ribose sugar, and this X here is supposed to represent the nitrogenous base. So it can be any, nitrogenous base. So that's just represented by an X because it would take up too much room to show graphically. With the cap, there is a couple variations that you can have. You can have cap 0, cap 1, and cap 2. So in cap 0, you just have the methyl on the 7 of the G of the cap. That's cap 0. Cap 1 is you have a methyl on the second position of the first nucleotide ribose as well. So if you look here, this is the ribose sugar. Each of the different positions on the ring has its own number. And this right here is the second position. So you have a methyl group there. So in cap 1, you have a methyl group on the second position of the ribose sugar. And on cap 2, you have one here, but also on the next nucleotide down the line, the second nucleotide. So it's cap 1 for the first nucleotide, cap 2 for the second nucleotide, cap 0 for it's not on any of the nucleotide, it's just on the cap structure itself. Yes?
And 7G always has the methyl group?
Yes. So yeah, Alan asked, does M7G always have the methyl group? And that is correct, yeah. If the process goes correctly. So this was just talking about the structure of the cap, but next we're going to get into the process of how the cap is made sequentially, and you'll see like which enzymes specifically are responsible for like which one's putting the methyl on the cap, right? Like, so theoretically, if that enzyme wasn't working, maybe you could get a cap that didn't have the methyl group, but who knows why that would be useful or in what circumstances that would be necessary. So, or what deleterious effect that could have. I don't know. So cap synthesis. So there are a couple steps associated with cap forming. It's just a little matter of memorizing them. But to walk you through them and have some pictures along the side. So the first enzyme in this journey is going to be RNA triphosphatase. When the 5' of your mRNA is being excreted out the polymerase, there are 3 phosphates on the end. That's just kind of how the end of RNA tends to be. There's the 3 phosphates there. So this triphosphatase enzyme is going to come in, and it's going to cut off the 3rd one on the tip. So the reason, triphosphatase, RNA triphosphatase, the name of what it does is kind of in the name itself. It's an enzyme because it's ACE that acts on the triphosphate of RNA. So here in the 5' end, it's going to cleave it and turn it into a diphosphatase. So now there's only 2 phosphates on the end. So that's step 1. Step 2, now you have another enzyme. This enzyme is called guanyl transferase. So it's going to take a GTP molecule. It's going to find one and take it. And we know that GTP is guanosine trisephosphatase. And GTP has a number of other uses that you learn about in biology. But it takes one of these GTPs, think like G protein cycle, and it's going to turn it into GMP, guanosine monophosphatase, and then it's going to take, so now we have 1 phosphate on the guanosine and 2 on the cap tip right now because the triphosphatase took that out. So it's going to then make this 5' triphosphate linkage between this little capping GMP and the tip that is now receptive thanks to the RNA triphosphatase. So now they're merged together. And then the 3rd enzyme that's involved in all of this is methyltransferase, which its name is also extremely spot on because it's going to transfer a methyl group onto our cap. Just praise be the Lord that they all kind of make sense, because sometimes they don't. But yeah, the methyltransferase is going to act on that N7. It's going to put the methyl group on. And then the fourth optional step is if you have a cap 1 or a cap 2, you'd have to methylate those as well. And that would be done by methyltransferase as well, because it transfers methyl groups. It's really good at that. Some would say that's kind of its one-trick pony. So yeah, capping graphic steps. So this is an image that's in the PowerPoints. It's basically just a recap of everything that I've said in graphical form, but to run through it just to interpret it and also kind of see it again. You start off with your RNA. It has 3 phosphates. RNA triphosphatase makes that 2 phosphates. Guanylyl transferase takes your GTP, makes it into GMP, and does that 5' triphosphate linkage. So now the GMP is on the diphosphate end. So now you have a triphosphate bond, which is what it's supposed to be. And then this says adumet, but that's a typo. It's supposed to say admet, meaning admethionine, which is done by methyltransferase, where we have this M7 added to the end. And then optional step for cap 1 and cap 2, you can add more methionine. You could add it to the first nucleotide, or you could add it to the first and second nucleotide. You can't have like a cap where there's just the second nucleotide and not like the first nucleotide. They go sequentially like that, to my knowledge. But biology is weird, so who knows? Maybe there's like a weird case where that could occur. But for our purposes, no. All right. So guanylyl transferase associates with the RNAP2 CTD. So this is going to be a big thing throughout this chapter. Well, I wouldn't say big thing. It's like a moderately sized thing that you just always have to keep in mind, that the RNAP2 CTD, RPB subunit 1, it's going to be helping out that C-terminal domain of the polymerase, is going to be helping facilitate capping, splicing, and polyadenylation. The way that it helps in this case is that the guanylyl transferase is associating onto this RNAP CTD. Moving on. So now let's talk about why we have the cap. What the heck? Why? Who needs a cap? I mean, people don't need hats. It's like a fashion thing, but you don't need a hat. Well, RNA needs a hat. And there are these 4 reasons. The first one is to protect the mRNAs. So the enzyme RNase is just around. It's just around all the time. You can never get away from it. RNase is responsible for degrading RNA. It's in the name. What happens is, if you have a buffer region on either side of the RNA, a cap and a tail, the RNase degrades inwards to the middle of the RNA. So if you have a buffer on each end, it's going to make it safer for the RNA. It's going to take longer for the RNase to degrade the RNA if it comes in contact with it. So it makes it more stable by having like a little buffer length of nucleotides or something on the end. That's kind of a protectant. Another reason is it increases translatability. And later on, there's like a little graph in the PowerPoint that shows the relationship between caps, tails, and translatability. We'll get to that. But just by having a cap, it increases the efficiency by which proteins can be made off of the mRNA. It also helps with transport. So by having a 5' cap, it'll be easier to transport the mRNA out of the nucleus to the cytoplasm, to where it can then interact with your ribosomes. And it helps increase the efficiency of splicing by having a cap, but particularly only with the first exon, or the first intron, sorry. The cap only helps with splicing what's directly next to it, and the tail does the same thing. It helps with splicing, but only the intron that's directly next to it. But it does moderately increase the efficiency of splicing, so it kind of like that little side benefit. So we'll talk a little bit more in depth about these reasons why we have caps and tails. Because spoiler alert, the reasons that we have caps is kind of the same reason that we have tails. Just it's pretty much the same reasons. We'll go into it in a little bit more depth when we get to tails. So now to go over what you want to write down for an essay along capping specifically. So in terms of like essay topics, from last chapter, I could really only think of one possible topic, which was the spliceosome. Like that could all be a huge essay, but this could be another one. Capping could be just a whole essay by itself. Polyadenylation could be another whole essay by itself. But for the capping essay, if you get that, because I know it's like she gives you some options and you have to pick. Capping is definitely probably likely one of the essays you'll see. So know when it's happening, when the first 30 nucleotides are jutting out of the polymerase on that 5' end of the RNA, where it's happening. So this is in the nucleus, and it's happening at the same time as transcription. Why it's happening. So those 4 functions that we went over, and kind of the general idea behind each one and why. And then how it's working. So explain those 4 steps, and be sure to pay extra attention to the 5' triphosphate linkage, and touch on that. And also, it wouldn't hurt to touch on the role of the CTD and how guanylyl transferase associates with the CTD to help with capping. So you could take pretty much everything I've said up to this point and turn it into an essay, and we've been talking for like 20 minutes. So now we've successfully beat capping to death. Now let's move on to polyadenylation. Polyadenylation is a bit more involved of a concept than capping is. Capping is a little bit straightforward, but there's more information on polyadenylation comparatively. So it might be like a little bit harder of an essay topic if you had to pick between the two of them. The general concept behind polyadenylation is relatively straightforward. It's just this process by which we add on A residues to the end of an mRNA. So here's a little graphical depiction. You can see our 5' cap, and then here's our poly A tail. So poly A tail, name is very apt. It's just a bunch of adenosine monophosphate residues. So it's an adenosine, a phosphate, adenosine, phosphate, because they're phosphodiester bonds binding them. And we call it the poly A because there's a lot of them. Generally 250 plus A's. And it's on the 3' end of the RNA. So we know that the RNA is being built in the 5' to 3' direction. So the tail is going to be one of the last things that's being made on the RNA. But it is important to know that the A is not something that's in the DNA. It's not, there's not like a bunch of T's in the DNA code that then encodes for A's. You have your coding region, your protein ends, and then enzymes come in and add on the poly A tail. It's not something that comes from the DNA. You know, we call it post-transcriptional modification. That's kind of what we call these things, you know, capping, splicing, poly A adenylation. We call them post-transcriptional modifications, but that's a little bit incorrect because it's actually co-transcriptional modifications, if you're being pedantic about it, because they're kind of happening at the same time as transcription, and we'll talk about that towards the end. But the point is, just know that polyadenylation it's not in the DNA. We'll get to the steps as to how it comes into being in a little bit. All right, so polyadenylation, we know, is this process by which you put A's onto the end of your mRNA. So the enzyme that does this, because like I said before, it's not done through the DNA code and translation or transcription. It's done by an enzyme. outside of that. And the enzyme that does that is poly-A polymerase. Aptly named again, it's polymerase. It adds on polyase, or PAP for short. And there are two types of PAP. So there is nuclear PAP, which is responsible for adding on your initial 250 nucleotides of A in the nucleus. And then there's cytoplasmic PAP. So like we said before, RNase, ever present, always breaking down RNA. And so you have this cytoplasmic PAP that exists there to continually add on A's to the poly-A tail to keep the RNA still working. And the reason this is because RNA is not a one-use item. You actually use a single RNA multiple times to translate proteins over and over again. So by having the increased integrity of your RNA, you can increase the translatability by getting more cycles of protein production out of it. Yeah, and then so something to note, once the poly-A tail is gone, the RNA molecule is pretty much donezo. It gets degraded. So the length of the poly-A tail can then control gene expression, right? Because like if you think about it logically, the longer the tail is, the more cycles that RNA could undergo translation, which means the greater the protein yield. And by modulating the length of that tail, you can increase or decrease gene expression, which is really fascinating to me. All right, so the functions of the poly-A. So pretty much the same as capping. Protection from degradation, we just talked about. Translatability, by being more stable and having a longer poly-A tail, the protein can be made multiple times. But beyond just that, it increases translatability because a longer poly-A tail helps association with ribosomes. So it's kind of this double whammy of like, yeah, you can do more rounds, you also will get a better affinity for ribosomes. It helps with splicing in the last intron, kind of like how the cap helps with splicing in the first intron. And stability, pretty much said that. It seems kind of redundant, protection from degradation and stability. It's a little bit redundant. To give more lip service to that aspect of it, it enhances what you could say is the half-life of mRNA, is a way you could say that. It can be used repeatedly by the ribosomes. But another little fascinating part, and this is important to note, I would definitely include this in the essay, is that it actually helps with RNA circularization. By having a poly-A tail, you can recruit these binding proteins that are able to then link the cap to the tail and circularize the mRNA. And this is more stable than linear RNA because it prevents the RNase enzymes from easily accessing the ends, which is where they work off of. So now we have our next player in the poly-A story, and that is poly-A binding protein 1, PAB1. The purpose of this enzyme is that it binds to the poly-A tail and it increases the translatability of that mRNA. So in the last slide, I talked about how having a longer poly-A tail increased association with ribosomes. The reason that is because poly-A binding protein 1 is then able to latch onto that poly-A tail and increase its translatability. So it helps in that process. It's not the only thing that affects it. Just longer poly-A's in general just associate better with ribosomes. But this is an actor in the story that helps in that regard. Now, just generally speaking, the effects of cap and tail presence on translatability. So I was too lazy. Yeah, I was too lazy to put this graph in, so I'm just going to move to slide 14. But yeah, there's this little graph here. So if you have no cap and no tail, this is your translation in terms of protein synthesized. So you can see you have really low amounts of translation with no cap and tail. With a cap and a tail, you get a lot higher amounts, And if you're missing either a cap or tail, you're kind of somewhere in the middle. You're not quite getting. So now let's talk about the effect of poly A length on translatability. So this is slide 15. So the longer the poly A tail, the better association with polysomes, which is just a cluster of ribosomes. I think if on your essay, if you didn't want to have to bother to memorize that little detail, you could just say poly A length increases association with ribosomes. That's not an incorrect thing to say. But you can see here, the biggest jump is between 5 and 30 nucleotides. Past that, you start to see diminishing returns. But yeah, between 5 and 30 nucleotides, that's when you see your best returns on poly A translatability lengthwise. Past going 30 nucleotides, that's more a function of increasing stability. All right, so poly A signals and poly A site. So we're getting closer and closer to talking about the steps. But before we start getting into those steps,
She considers 4 steps. She now considers this step 1.
yeah, this you would have to kind of talk about this as a prelude to talking about the steps. It's kind of like, you know, it's a story, right? Like the process is itself a story. You have actors in it, which are like your different protein and enzymes. There's the signals, and you kind of have to explain each individual part. So then when you go into the steps, you're just kind of referencing something you already said. It's a little bit easier if you're like, okay, so the things involved are this enzyme, this enzyme, this enzyme, and these sequences. So then you get into your steps, and you're like, okay, so this enzyme I mentioned earlier comes in here, does this, and recognizes this sequence that I talked about. Or like, you know, it's up to you how you want to structure your essay, but that's how I think about things, and it makes it a little easier for me. So that's kind of why the Quizlet's set up this way, where I'm introducing each part before going into the steps. It gives us some context. But here is your general mammalian polyadenylation site and other associated elements. So your poly A site is typically in the middle. Upstream of that is would be this consensus sequence, which is just called your poly A signal. The consensus for it is AAUAAA. And the closer that is to consensus, the better rate of polyadenylation you'll get. So this consensus sequence has a very important impact on polyadenylation efficiency, and you can see that in this graph here. So this last one here is your consensus, and you can see very high amounts of efficiency in polyadenylation. But as you start to do mutations to that, you see decrease and decrease. So that's the very important part of the signal. And then there's just this downstream GU-rich element, followed by like we just kind of lump it together with the GU/U element. So there's like usually like a GU, and then the longer it goes on, it just starts to change to U. Those are your basic parts of this poly A signals and site. So the poly A site, yes, I got it.
Followed by 24 base pairs later by GU, which sequences at 24 base pairs after the polyadenylation site or the AAU sequence.
Question. So the question he asked is when it's saying upstream, it's upstream in relationship to what? So if you think about like start site, right? It's usually in reference to a single like nucleotide position. That's like your +1 site. So what I am going to assume is probably the poly A site specifically is a single nucleotide where it's like after that site, that's where your poly A tail is going to be added on. So that'll be your like the one nucleotide. Then probably from that single nucleotide 20 upstream. So like if we can visualize that as being like the point right here, this way up until you start hitting green, that's 20 nucleotides. And then from that point downstream until you start hitting the GU is probably 24 nucleotides. That is my best educated guess as to what I think it would be, and I would have a difficult time thinking it would be anything else than that unless the poly-A site is multiple nucleotides long, but that doesn't make any sense because the poly-A site is just like where polyadenylation starts, so it would just be like a single thing, right? But I don't think that's going to be that level of detail necessary for the essay. I think probably even the exact nucleotide distance these elements are away from each other is like a lot of detail. Like you can go ahead and memorize it. Like it's not going to hurt to put on the. I think as long as you say there's this or this AAUAAA, and then in the middle of that's the poly-A site, and then downstream from that is the GU/U element. And I think as long as you know that those are the players here, I think you're fine. But moving on. So now we start getting into the steps here. I'll do this overview of polyadenylation first, and then I'll get into like the individual steps in detail. So I'll go a little out of order with the Quizlet's doing. So the first step in polyadenylation is that you have your pre-mRNA, which is your growing transcript, and it's cleaved at the poly-A site. And that's the cleavage step, so that's your first step. After that is initiation, then elongation, and then termination of transcription. Initiation and elongation are simpler steps than the cleavage. There's less going on with them. So pretty much everything that you kind of need to know regarding initiation and elongation is just on this slide already. Even though this is kind of like the simple overview, there's not really too much more depth on the PowerPoints that Dr. Daud's going to need you to know. So having said that, I'm going to backtrack to this cleavage of pre-mRNA step where we go into the specifics of each factor and do it a little bit closer detail. Starting off, let's list our actors. So we have the following proteins involved / enzymes involved in this process. So you have your CPSF, which is your cleavage and polyadenylation specificity factor. This is going to be responsible to binding to that AAUAAA sequence. Then you have CSTF, which is your cleavage stimulation factor. And this is going to bind to that GU-rich region. And then you have your general cleavage factors, which are CF1 and CF2. And then obviously you're going to need your poly-A polymerase. It's going to be involved in this polyadenylation process like we talked about before. And you're going to need your PAB2 is also going to be part of that. We talked about PAB2, did we? No, we talked about PAB1. Okay. Back here we talked about PAB1, which helped increase translatability by binding onto the poly-A tail. That's PAB1. Yeah, not a part of cleavage. That's PAB1. Now we have PAB2, which I guess is a new guy that we haven't talked about before, but is related to PAB1 in some way, same class of protein factors. All right, so now that we have our various like enzymes and stuff laid out, let's go through the steps. So in step 1, your proteins bind and associate with the RNA poly2 CTD. That's going to be the first thing. So the factors that I listed up top, they're going to do that. So after you have your proteins associating with the CTD, your CPSF, the cleavage and polyadenylation specificity factor, that's what's going to cleave, and it's stimulated by the CTD right there at the polyadenylation site. All those enzymes, all those cleavage enzymes that I listed above, they're all working together to do this, but I'm guessing just CPSF is like the one that actually does the enzymatic reaction to cut it. But all the cleavage factors are obviously assisting in the cleavage. And it's the association with the CTD that's going to be like stimulating the cleavage to actually happen. All right, so now you have cleavage happen, right? So cleavage is really the first step. So you have to. It's kind of not as neat as like the mRNA is being transcribed, and then when it runs out, then it just falls off. It's not quite like that. There's like a continuous feed coming out, and the polymerase stays on, and it keeps making more. And once you get to the end, rather than the polymerase stopping, the polymerase just keeps going. And you actually need the factor to come in and say, okay, here's the end of the transcript, and then you cut it. But the polymerase is just going to keep making more and more. The mRNA like falls off once it's been cut, and that's the end of the transcript. But the polymerase would just infinitely, go on. It's through the process of termination that we have to stop the polymerase from just keep making stuff. We'll get to that. So once your PAP binds, yeah, go ahead.
Was there a certain order that we need to know?
Of the factors, the cleavage factors?
Yeah.
In this diagram, though, because I was looking at the PowerPoint earlier today, and all I was saying is just that there's no factors, and then immediately after that, all of them are just there. I don't think, in the real world, in like practice, factors don't bind like one after the other. Like when you actually learned about like the subunits associating in RNA polymerase 2, right? They initially taught it to you in a very specific order, but it wasn't like that again. It's a hollow enzyme. All of the factors associate together, and then they come on to the promoter. I think it's a similar case here, where it's just like all the factors are there, they just kind of come in together. You can think about it like that. That is most likely how it is in real life. Yeah, if she didn't say, just think of it as they all come at once. All right, so after you have this cleavage done by CPSF and the other cleavage factors, you have your cleavage factors dislodge. They dissociate from all this, and then you have your PAP, polyase polymerase, bind on. And poly-A polymerase is going to do what it does best, which is the only thing it does, and it's going to polymerize A's. So it's going to add on 10 nucleotides. So specifically the factors that dislodge are CSTF and CF1 and 2, but CPSF stayed on after the cleavage, which was also the guy that did the cleavage, right? After it cleaves, it stays on, and it's going to help out the poly-A polymerase. Doesn't say how it helps it out, it just says it does. So the job of poly-A polymerase is just to make those first 10 A's. That's its job. After the first 10 A residues are made, PAP goes, and now PAB2 comes in. Now PAB2, poly-A binding protein 2, not to be confused with poly-A binding protein 1. Wait, did I say PAB leaves? Sorry.
I just read that.
Yeah. All right, PAP doesn't go anywhere. PAP doesn't go anywhere. Yeah. PAP doesn't go anywhere. But on his own, he trucks through. He does the first 10 nucleotides. But after that, then you have PAB2 come in, and it's going to help out PAP to take it down to 250. Sorry. Bear with me, Alan. I'm so sorry.
No, you're good. I just saw that too. I read that on
Yeah. Those are pretty much all of the steps. This is pretty much everything for polyadenylation. What's up?
So I'm just making sure when it comes and cleaves the messenger RNA, there's still transcript messenger RNA after, because it's continuing to transcribe.
Yes.
RNA polymerase.
Yeah, and I can show you a picture of that. Yeah.
Okay.
Here you go. Here's a picture of that. So he asked about like after it gets cleaved, the polymerase is still there, it's still working, and so there's still transcript after where your mRNA would be done. Yeah, kinda. So right here is our cleavage site, our polyase site, and the polymerase is still here on this DNA, and it's still making transcript. And then there's going to be a mechanism here that's going to allow for the polymerase to stop making transcript, and it's going to degrade here. I'm going to get to that though. We're not there yet. But yeah, you're correct, Alan. All right. Sorry about that little mix-up. I didn't realize. CTD code. Now I was kind of just talking about the role of the CTD of the polymerase in this whole process. Basically, it's just that I kind of already said it in the beginning. So the CTD helps with all three of these processes. They're all three happening co-transcriptionally. The shift as to like, is the CTD helping with capping? Is it helping with splicing? Is it helping for polyadenylation? Is based on the phosphorylation state of the CTD. So as transcription is occurring, and since this process is happening co-transcriptionally, as transcription happens, the CTD of the polymerase is going to accumulate phosphorylations, and that's going to like phase transition it from capping to splicing to polyadenylation in a kind of a smooth sequential manner. So that's the CTD code, and to show a little picture of it on your PowerPoint, you can see here fewest amount of phosphates on slide 31. You're in capping still. Accumulate some more phosphates. Now you're splicing, and then you have the most amount of phosphates, and you're cleaving. All right, so this was a little side note. There's not like a whole slide on it in the PowerPoint, but this was like a detail that Dr. Doud harped on in the lecture, so I made a dedicated slide to it. I think there is some of it in the slide. Transcription termination is closely linked with polyadenylation. The two processes are intertwined. In order for transcription termination to happen, you absolutely need the cleavage factors that we talked about already. The cleavage factors are responsible for cutting the mRNA, which is necessary for it to end. After that, there's some additional steps. And as far as transcription termination is concerned, it doesn't, within that process, that is the point at which these two processes stop being related to each other. You need the enzymes, the cleavage enzymes, to cut the mRNA. That is needed for transcription termination. It's also needed for polyadenylation. Once the transcript's been cut, now the processes can finish up in their own, they can diverge. That's kind of like a fork in the path. Transcription termination couldn't be concerned less with, does the mRNA get its poly-A tail? That's polyadenylation process's job to do. So, but in order for transcription termination, you need those cleavage factors from polyadenylation. So I've just repeated the same thing like 5 times, but just to drill that point into the ground. So now we'll talk about termination of transcription. So we're getting close to the end of the chapter. It's just one last little thing. Once CPSF cleaves at the polyase site, it releases your growing mRNA transcript. So I'll zoom in here. Actually, yeah, there we go. So CPSF cleaves here. And then you have in the DNA code something downstream from that poly-A site called the COTC site. So what this is it's going to cause the polymerase to transcribe a length of RNA in this kind of growing, like still going on transcript that has self-splicing activity. So after the cleavage has happened around here, Then you have your COTC site, and it's going to cleave itself. And by cleaving itself, it's going to allow a process called torpedo termination. In terms of order of sequence, first you have that COTC, the COTC ribozyme cleaves itself, and then you get torpedo termination. Don't think too hard about how that transition happens. There's not really a good explanation in the slides. But then you get torpedo termination. Once you have step 1 and 2, so you have the cleavage and then the COTC cleaving itself, those two steps together are going to signal your RNA polymerase II to dissociate off of the DNA template. And then you have torpedo termination, which is the last slide. So this is a really simple concept. Essentially, here at your COTC site, there is this enzyme called XRN2, which is going to come onto where the COTC site cleaves. So by the COTC site cleaving itself, you have this kind of noodle of RNA that is no longer doing anything. But this acts, by self-cleaving, it acts as the entry site for the XRN2. Why the XRN2 couldn't just start right here, I don't know. It's not really clear to me, but it's not that deep. Past that, XRN2 just starts eating the noodle, and it eats the noodle until it catches up to the polymerase. Once it catches up to the polymerase, it forces the RNA polymerase to dissociate. You can see. So it eats the noodle until it gets to the polymerase, and then it kicks it off. And that's how transcription ends. So to recap, everything in this chapter has been on eukaryotes. And if you think about it, it's because eukaryotes have mRNA processing. Prokaryotes don't have mRNA processing. It's just the genes transcribed and translated into proteins without any processing. So all of these processes are eukaryote specific. And so like transcription in eukaryotes versus prokaryotes is different, just in general. Like the steps are completely different. So that's going to be the end of chapter 15. Alan, do you have any? Oh, hey, Chrissy, I didn't realize you were here.